Hello.
Hello, dear participants. I welcome you all again to session 11. It will be conducted by Professor Anjana Pandey. It is also a lab session entitled as Use of Software and Analysis of Results. Over to you, ma'am.
Hello, participants. Are you back? Hello. Do you have any query?
Yes, ma'am. We are
Jen,
back.
Okay, that's nice. Should I start now, na?
Yes, ma'am.
Any doubt in the previous laboratory?
No, ma'am.
Okay, that's good. So I'm disabling you again. Fine. So in this laboratory session, uh, I'll be explaining you the other bioenergy production, which is can be extracted using the algal biomass. As I explained you in my lectures also, that algal biomass, which is considered as a third generation feedstock, and it is a very potential feedstock in a sense that it is sequestering the CO2 from the atmosphere and getting fixed in the form of the glycogen. Along with that, lot of pigments, and they synthesize oils, and that is a reserve which is present in different algal species in different amount in natural condition. So our objective is to extract these oils from the dried algal biomass, which is present in surplus amount in nature everywhere. So for that, the first step is the harvesting of those algal cultures and then drying it, followed with the extraction of the oil. And after these oils are extracted, these residues can be hydrolyzed to liberate the monosaccharides, and those can be used for conversion to bioethanol or biohydrogen or biomethane or hethane for commercial use. And the waste product or the byproduct liberated can be used for soil as fertilizer. So it is such a useful feedstock. So I thought that you all must know how to process it. So I am playing this video so that we can proceed ahead with it. This has been prepared by Priya. She is working in the area of biohydrogen production and the pilot plant as well as technology development. She used the immobilization and the use of various nanoparticles for augmenting the hydrogen production. This work which I am presenting has been done by one of my MTech student, Ms. Prajakta. If you see the overall status of the biodiesel production by the state, which is in million gallons per year, you can see that across the different states, among those, Iowa, you see, Texas, it is all in USA. Iowa is leading in comparison to the other states. Similarly, if you see in the India, then you will see that year-wise production of the biodiesel from 2011 to 2020, we are not producing it at a large and continuous rate, but it is okay. We need to improve it, and for that, again, I'll say that the initiative from the government has to be taken up. I told you that in the policy it has been decided that 5% blending of biodiesel is recommended in the diesel which is being used. However, the biodiesel production market it has increased from $82.7 million in 2011 to $185.3 billion in 2021. So you can say that there is a steady growth in 2016, but a rapid increase in production between 17 and 21 due to higher oil prices. So it has been decided that it has to be increased by 65.7 billion gallons per year by 2021. Look at this detail that in the first 3 quarters of 2018, the Indian biodiesel plant produced about 15,200 metric ton of biodiesel during the plant upgrade construction. After the expansion, current Kakinada plant capacity is increased to 165,000 metric tons of biodiesel. It is one very important achievement of India. If we compare the production in global market, then you see that it has been predicted from the expert report that the market of biodiesel production has increased from $82.7 billion. It is again repeated, the same slide. Just hold on. It is just a pic that how biodiesel plants are constructed in different area. Okay, not required to see these diesel plants. So coming to the laboratory, that is the it is a marine algae. It is a microalgae. It is small in size, very small in comparison to the Anabina and Microcystis, Spirogyra. So it has been exploited for extracting the biodiesel and using the two-stage process of optimization method. So someone has asked me in the morning how to select the parameters for the optimizations. So the same way here we did the two-stage optimization. In the first stage of optimization, nutrient supplementation, where media constituents were optimized, and for that F by 2 media is used, and this F by 2 media is actually a simulated dairy wastewater. It's just like cheese whey which is being collected from the different port where milk is processed for making the cheese. And in the second stage, different stress factors which are nutrient starvation, salinity and pH. Salinity, you all know that it is the concentration of the salt, and nutrient starvation leads to the production of more lipids. So these were employed for optimizing the factors using response surface methodology. The way you did in the morning. So here the media is first of all you have to optimize. For that autoclaving you have to do because it is a controlled condition experiment. So naturally you have to use the autoclaved media. And these autoclaved media is kept in the UV chamber along with the other useful things. The UV chamber where it will be exposed for UV for 15 to 20 minutes. Okay. So all must be surface sterilized using 70% methanol. The action of methanol is more in comparison to the ethanol. Remember, everyone, the 70% methanol gives you better sterilization. So don't go for the ethanol. Use the methanol. It is much better. So it keeps the sterilized condition because it has a more contact time to the surface. Reason behind this is that it has more time. It remains to the surface for more time in comparison to the ethanol because ethanol is more volatile in comparison to methanol. So it gives the sterilization better. And then the algal culture which you want to use for extraction, you can take and start working. But before starting the experiment, always switch off the UV, keep it for 10 minutes, then only you open the hood. Initially, the nitrate concentration in the sample is measured by ultraviolet spectrophotometric screening method. And for this, 5 ml of sample is taken and 100 microliter of HCl 1 normal is added into it and absorbance is taken directly at 220 nanometer to obtain nitrate and also at 275 nanometer because absorbance is 275 nanometer gives the interference of dissolved organic matter. And if you get the absorbance at 275, then you have to deduct the absorbance which you achieved at 220 nanometer. So that will be the nitrate estimation which is not contaminated with any of the organic matter. Okay, so we did the subtraction for 2 times and it was compared with the standard curve to know the exact value of nitrate in present in the medium. Looking for the another factor estimation which is phosphate estimation, and this is estimated by another method which is called as ascorbic acid method. So 5 ml of sample is taken in a clean dry test tube and 800 microliter reagent is added and mixed thoroughly, and the absorbance is measured after 10 to 30 minutes at 880 nanometer. Always put the blank because it gives the some absorbance if you obtain at a specific wavelength that you have to subtract from the absorbance which you achieve, and then exact concentration you obtain by comparing it with the slope value which you obtain by drawing the different concentration of the standard solution using the standard curve. Now coming to the one very important parameter which is called COD estimation. Everyone is speaking about that. This is chemical oxygen demand and this is measured by open reflex titration method. For that you have to take your sample in the COD vials and 1.5 ml of potassium dichromate reagent is added, followed by addition of 3.5 ml of sulfuric acid, and then these vials are capped properly and put in the block digester which is preheated and kept for 2 hours. So after completion of 2 hours, vials were cooled down at room temperature and then solution is transferred to the conical flask where one has to add the ferroin indicator and then it has to be stirred rapidly over the magnetic stirrer and it is titrated against the standard 0.1 molar FAS solution. This is ferrous ammonium sulfate, and then at the end point you see the sharp color change from blue green to red brown. In this case also you take the distilled water as a blank Okay, and you have to do the calculation as per the formula. The detail of the methodology, everyone should follow the AFA protocol, APHA. You will find out using the Google. So what we chose the tetracelmis growth on the simulated dairy wastewater, where 12 gram of Anik spray milk powder is dissolved in 1 liter of distilled water, and this solution is called the SDW. So we have chosen three percentage. One is 25%, 50%, and 75%, and the 100%. To see the effect of the F2 media, which is having different concentration of the milk powder. So after cultivation for 10 days in 250 ml conical flask containing 100 ml media, cells are harvested using centrifuge at 5000 rpm for 10 minutes at 4 degrees centigrade. And then it is dried, and weight of the dried mass was calculated using gravimetric analysis. Further, COD reduction and growth rate was analyzed with each dilution, for each dilution, using UV visible spectrophotometer by taking its absorbance at 650 nanometer. Okay, so this was the first optimization, that what concentration of this simulated dairy wastewater to be used in your experiments. So for harvesting, you can see it is centrifuged, and pellet is obtained at the bottom. It is a fixed rotor centrifuge. So you can see that here centrifugal force is sedimenting the tetracelmis biomass. Okay, then what is she doing? She is trying to all these harvested biomass will be aseptically extracted. But it was not needed to do the aseptically, let me tell you, because after all you have to extract out, and the residues will be used for extraction of hydrogen. Let us see what is she showing. She is opening the lid of the petri dishes, and she's putting some labels over that. This time she is using the glass petri dishes. You learn how she handling. Learn the handling. This is very important for you to understand, because you are not here, no? Okay. She was spraying the 70% methanol probably. She's disinfecting the surfaces, open surfaces of the lids. Let's see what is she doing. Okay. She's opening the lid of your centrifuge tube, which is commonly known as the Falcon tube. Now she is dispensing of the supernatant. She will do for all. Then what is she doing? Then she is saying that supernatant were used for biohydrogen production. In the oven, I think she is going to dry it. Yes. So all the wet biomass which you got, you have to dry it in the oven, and usually 70 to 80-degree temperature is used for drying. The lid is covered with the aluminium foil, and dried mass is then scraped off from the petri dishes, and then she will collect all those biomasses, and then she is transferring that biomass in the another falcon. Okay, this is how it is being used. Okay, she just wanted to tell you one should know the idea of doubling time of the microbe which we are using to study the impact of different dilutions. So different dilutions we prepared for the media, that is 25%, and then this is the control. This is the 25% red one, and this green one is the 50%, and magenta one is the 75%, and then for 100% this is this one. This you can see the impact of dilutions on the growth. Yes. Then after that, for second optimization, 4 stress factors are chosen: sodium nitrate, sodium hydrogen phosphate, salinity, and pH. So in this case, 4 variables and 5 levels are chosen. Why? Because it is going to be done using the CCD. According to the CCD, equal distance was chosen, again to study the impact of linear effect as well as interactive effect of different levels of variables with among the set of reactions. Here you see that there are 30 runs which we used for studying the 4 variables using the CCD. Since in the CCD you have the 5 levels, that is why number of runs have increased to 30. So what actually your objective is to optimize the dry cell weight. More biomass, it means more substrate for biodiesel production or hydrogen production. In the first stage, biodiesel production. In the second stage, biohydrogen production. Okay. So here you see that this is the dry cell weight which has been achieved in different runs. In different runs you can see. And this is the predicted value which software can give you, or if you want you can calculate using the equation which you obtain through the model. So this is the model equation which you obtained, and here you have to put the value of A, B, C, D for each run, and then using this equation you can predict the value of the lipid content, because your objective here is to increase the lipid content. You can also go for the dry cell weight also. You can also go for the lipid content. So there can be 2 responses. It is up to you what you want to do. More biomass, naturally more lipid, and So data analysis by ANOVA of CCD shows that it is highly significant. That is why 100% of the experiments are close to the model which is being predicted. And F value is too high, that is why it is 100% significant. And P value shows that 0.0000 up to 3 decimal place. That is why not a single experiment is deviating from the model. And lack of fit, here you see its F value is too less, that means it is not significant. So here you see that for all the 30 runs, we have plotted a graph to show you the variation in the lipid content, and so you can visually see that how the combinations of different parameters are affecting the yield of lipid. So I think now you agree that this model, whether it is CCD or BBD, that will help in optimization of the process. So optimization of the process is one of the very important aspect which should be done in the every laboratory for their ongoing research. Whatever you are doing, you must get the result at its optimum before it is being reported or industrially exploited. And the surface plot at a glance gives you the result and showing the interactive effect of two parameters at a time on the response. Like this light region here is showing that at this pH here and at this phosphate it is showing optimum. This is showing, if it is showing here, you can see just it is at the center, equal contribution of both the factors in controlling the lipid content. Here you can see that phosphate is phosphate and pH, if you see, pH is controlling and phosphate is also controlling, but the contribution of pH is more. And here you can see again the contribution of both the salinity and nitrate is equal for controlling the lipid content. So this response surface plot is very useful. At a glance you can know what is the result, what is the impact, so you can immediately decide, yes, these are the factors which are regulating the process. So whenever you use any bio waste, so you should analyze in that bio waste how much nitrate is there in it, how much phosphate is there, how much sodium is there, sodium chloride. So what will, because it is ultimately going to affect your biodiesel extraction. And that in every industry we have the analytical laboratory. That is why it is needed. So because you know that when you go and from different farm to different farm, the quality changes. Quality changes, it means their nutrition content is changed. Nutrition is changed, it means their nitrogen, phosphorus content is changed, because that is controlling the overall quality. And then its market evaluation is being considered based on its nutritive value. And then when you go for the optimization plot, it gives you the exact value of the optimized point of all the variables. So these optimized value gives you the point or the values which you use for validation study, like you did in the previous experiment. So after this you get the response, and that response, if it is close to the predicted value, then you say that you have validated the model successfully by experiments also. So the validation is also a very important step for every model. So after that, you have sufficient quantity of cells. So you harvested it by centrifugation at 5000 rpm for 10 minutes at 4 degree, and then you dried it for 2 days at 50 degree centigrade, and then it was blended using mortar and pestle. A lipid was extracted and transferred to the pre-weighed vials, because later on you will have to weigh everything. So then you can subtract the uh, pre-weighed vials weight from it. And then solvent was evaporated in water bath at 60 degrees centigrade for 2 minutes. And then it is dried and weighed using gravimetric analysis. And then by acid catalysis method, we produce the transesterified esters, and methanol is used for helping in the transesterification. So extracted amount of dried lipid was dissolved in 2 ml of chloroform and 2 ml of 2% sulfuric acid in methanol. So then it will be analyzed, all those biodiesel, the lipid, the biodiesel which we found in the form of the esters. We sent it for its characterization by GCMS, and then after doing the GCMS analysis, we got the plots also. I'll show you. So the solution which you obtain, it was incubated in water bath at 90 degrees centigrade for 40 minutes, and then 2 ml of distilled water was added and centrifuged to speed up the phase separation, and then lower phase was transferred into a pre-weighed vial and dried in vacuum desiccator, and that was sent for GC analysis. You can see here the peaks of the GCMS, which is showing the presence of different trans-esterified biodiesel components, and by name it is identified as at different retention time, like benzaldehyde 2,5-dimethyl. At 8.7 you get phenol. At 13 point something you got the hexadecanoic acid ethyl ester. So like that you obtained the different biodiesel components. So you see here that majorly the fatty acids which is present in the esters are either C16, C18 or C18 or C18 1. So this is showing the point of unsaturation. So finally we obtained that 75% dilution of the dairy powder was the optimal media for its growth. And then we found 51.65% lipid content was found in the optimized and validated experiment. And in that condition, percent COD removal was 95%. So based on these results which we obtained, we proposed that this tetracelmis could be a very promising organism for biodiesel coupled with bioremediation of dairy wastewater. So bioremediation, here you understand, remediation means removal. So dairy wastewater which is having the high COD, it is remediated. That means bioremediated through the by the tetracelmis, because it is utilizing the COD which is present in the wastewater, that is in the way, so it can be industrially exploited. Is that okay? Hello. Okay, fine. Let me allow for everyone. Hello, participants. Hello, all of you. Hello.
Yes, ma'am. Yes, ma'am. You are audible.
Okay. Do you want to ask anything?
No, ma'am.
Okay. I was telling you that nanotechnology has a very important or revolutionary technology, I can say rather, because I told you that whatever you are doing, whatever process you are developing, if you use the nanomaterials, it will altogether change the fate of your process. So if you people are interested, we can discuss something about the nanomaterials, its synthesis or characterization, if you wish. It's up to you. Tell me. Hello.
Participants, if you have any query, ma'am is present there for you. And if you have no queries, then we are about to end this session. Participants, please go ahead with your queries. Any doubt regarding any of the lecture presented by ma'am? If you want to ask anything extra regarding the title, please go ahead.
Hold on. Okay, participants. I would like to discuss some more things with respect to your laboratory. In that regard, I was talking about the use of nanomaterials. What I told you that whatever you are using in your medium as a salt form, that is considered as the bulk material because it's a salt, and that salt is not present as individual molecule. Let us say, for example, I'm talking about, say, ferrous sulfate, FeSO4. Okay, this FeSO4 you won't get as a single molecule. It is present in the form of the agglomerates, which you use in the laboratory, which you weigh and then use. And when it is dissolved, you get the ferrous ion and the sulfate ion. And then your microbe or your plant, they absorb it. Fine. And when I say that it is in the form of the nanomaterial, in that case, what happens? Usually, we prepare the aggregates of these reduced ferrous in the form of the Fe0. There is no plus charge, there is no negative charge, it is Fe0. And that Fe0 form is either present in the environment when it is being synthesized, it makes its oxides. Some of the oxygen go and chelate over there. So that is a cluster of few atoms, and when they are in that form, then their electronic configurations as well as the movement of the electrons, it is localized in the outermost shell, and they start moving and changing their positions. That's why they are free to move, because it is not present in Fe2 or Fe3 stage, which is stable. It is present in the nano form. So in that case, what happens? Their electrons, which is present in the form of a quantum, they show a different behavior altogether. So their electronic properties, their magnetic properties, as well as other properties, mechanical or conducting ability, everything changed. So that is why when these nanomaterials are synthesized and used in the bioprocess, they affect the bioprocess at different stages. So all those reactions which require electrons for its reduction or oxidation, these nanoparticles interfere there. And when they interfere, they affect the reaction in different ways. Either they either they provide the protection, or they lead to the free radical production. Protection, I mean to say, they capture the electrons and they protect the free radical to proceed ahead. So they just terminate the chain reaction of the free radical formation. Okay, so what happens? That results in the overall change in the process output. So in that regard, you always have a choice. Whatever process you are optimizing, you can think of using those nanoparticles of the related constituent which is present in your medium, or rather I can say the factors which are regulating the process. Reason being, these elements are present in protein in the form of a cofactor. So the interaction of these protein to nanoparticle will lead to the overall new scene. So the new fate of the process which you are regulating in the laboratory condition. So it also need to be optimized very carefully. And for synthesis, I would suggest some of the ways. Among those, you can go for the chemical synthesis way, or you can go for a green synthesis way. When you say it is a chemical synthesis way, then you can go for the hydrothermal way of synthesis. You can go for the salt to gel conversion synthesis. You can go for the co-precipitation way of synthesis. But you understand the basic of it. Whenever you are synthesizing those nanomaterials, you are either using the acid or base to change the oxidation state of the salt which you are using for making a specific nanomaterial. And when you are doing it, you can do you can synthesize it in various ways, either in the form of the dots, or in the form of the rods, or in the form of a various three-dimensional shapes which are available. But whenever you synthesize it, they are not synthesized homogeneously at the room temperature condition. For that, you need to do the calcination, and that calcination is the heating of that material which you prepared. And for that, when you heat it at a specific temperature for some period, some few hours, all the crystalline materials which you are synthesizing, they rearrange themselves. All the reduced element which is present, which we were talking about, to make in the form of a nano dimension. Because overall, we are reducing all the elements to its zero condition, and then we are stopping the reaction. So further, the nucleation side where the reduction has started, it is not able to grow further. So you are stopping the reaction by putting either new solvent into it, or you are putting in some stabilizer. So you stop their further growth. And then after you calcine to make it crystalline. Crystalline in a sense, all everywhere it has the same lattice structure. Whether you check at any corner of your material, everywhere it will have the same lattice structure. And if lattice structure is same, then your material, you characterize it using the XRD, X-ray crystallogram. You get the diffractogram, and that diffractogram gives you the specific peak. I will show you some of the peak which is being obtained by our student, so that you will understand. Just a moment. Okay, yes, this one. Can you all see this? This XRD pattern of crystalline nanoparticle. Like this one is being prepared for silicon dioxide, and this one is for cerium dioxide. You can see there is a very sharp peak, and these sharp peaks are showing the three digits over here. These three digits are nothing, these are the HKL planes of your lattice, which is present in the solid phase of your nanocrystal. And you get these peaks because of the constructive interference of the X-rays when it impinges on the different planes of your nanoparticles. And when they constructively interfere, they give you the signal, and that appears in the form of the peak. And these specific peak position at the specific theta value, it identifies the planes of your nanoparticle. So after this identification, you are supposed to take the UV, ultraviolet spectra, and using that spectra you get some peak in the specific UV region. So using the absorbance, one can make the plot between the energy and this X and Y value to obtain the top plot, and it helps in calculating the band gap energy. This band gap energy actually gives the difference between the energy difference between the valence shell and the excited level of your uh, electronic levels of your material. After characterization of this, one has to see the particle size distribution in the solution form. Because after all, whatever you have synthesized, you have to use it for practical applications, and for that you need a specific buffer or distilled water. So your particle needs to be first grinded properly, and then a specific percentage of that you take it, and then you sonicate it, and after the sonication you use the machine which is called particle size analyzer, where the specific laser is being impinged over the nano suspended particles in the solution, and that scattering angle gives you the overall particle distribution sizes. And here you see that more than 90% of the particles are of same size, but on the x-axis you can see the distribution here. If you compare these two slides, you will see that here the distribution of particle size is narrow, whereas in this case distribution is quite big. Okay, so this gives you the overall distribution of the particle size. This is study is called particle size analyzer, or you can say that it is the dynamic light scattering experiment. Okay, so this is the way it is the usual characterization. And after that, I'll share one more. Just wait. Uh, just hold on. Just hold on. Here you see. Look at this slide. Can you all see? Is it visible? Here you see that this UV spectra I was talking about, it gives a specific peak. This is the identification of the growth of nanoparticle. This is the one example which I am showing with respect to the gold nanoparticle. Here you see that whenever you are reducing this auric chloride, gold auric chloride is the sol of gold chloride, and when it is being reduced with the reducing agent, so those this oxidized form of the gold is reduced to the gold zero form, and then at the different nucleation site, as it is visible here in the TEM, this is the transmission electron microscopic picture of these gold nanoparticles. You can say at the nucleation site it is growing, and when it is growing you can see that various shapes of these nanoparticles. And this is giving you, when you see the close up, you will see that there is a different lattices and the planes of the lattices. So all the lattice characteristic can be obtained when you magnify it further. See, as you can see this, here you can see there is a quite wide distribution in the size, as it is clear using this particle size analysis distribution, which is your DLS study, dynamic light scattering study. So this is the way of characterizing your nanoparticle. This here you see that some of them are in the form of a rod. You can see here, this is the rod. And so, but in this case here you see the plot is also changed. And suppose if you are making silver nanoparticle, then the size and their distribution is different, shape is different, and the characteristics is also different with respect to the UV spectra as well as with respect to the size distribution. Suppose you wanted to go for the nickel oxide nanoparticle synthesis, then XRD shows you the HKL value, which is 111, 200. These are the specific phases of your specific nickel oxide nanoparticle, which is being compared through the literature to confirm that yes, nickel oxide nanoparticles are formed. And the UV visible spectra gives you the peak over here. Vandita, you see, the peak is visible, and that peak is considered as the point to calculate the band gap energy through the top plot. Here also you can see there is a wide distribution in the sizes. Likewise, zinc oxide can also be synthesized, and you can have the different HKL plane, which is similar to your JCPD database. Likewise, you have the size distribution in this case also, it is so wide. And then coming to the another one, which is graphene oxide nanoparticles, that is also showing the specific UV spectra as well as a specific identification peak, which is 002 for the graphene oxide. So you see overall that nanoparticle can give you the now you have the idea how it looks. This is, I am just showing you the way it is being simulated using one of the software to just let you an idea that graphene oxide is not looking like a gold. Here it is, you see it is a gold prism, but rather it is looking like a wafers, like a stage you can see, and over that gold can be attached, and over the gold any moiety can be attached, and these moieties could be nothing, it could be your proteins, the proteins which are facilitating the your bioprocess responsible for the conversion. I hope you understood it. So I will stop sharing it because it is the data of one of my students. So I just wanted to make you people acquainted with these new technologies and augmenting these nanomaterials. Overall, you can change the future and viability and stability aspect of your bioprocess, whether it is with respect to the bioenergy production or whether it is with respect to the sustainability, because you can engineer these nanoparticles, and engineering can be done by controlled and precise modulation, and that can be achieved by the controlled synthetic pathway of nanoparticles. I will again stop here to give you a break so that if you have any doubt, discuss with me, because it is for you people. So you have to ask questions. What you want, what is coming in your mind. Hello? Hello, participants. Any query? Hello?
Ma'am, no query from my side.
No queries. Okay. So I told you that after this, I would again go back to my practical session to explain you the molecular aspects of any bioprocess design. What I mean to say is that in my lecture, you might have seen one slide. I'll go back to that slide to elaborate the discussion, and in which, just hold on, I'm sharing it so that you will be able to link it. Yes. Can you all see this slide now? Is it visible? Here I told you that the use of genetic engineering, the use of synthetic biology, and the use of pathway engineering, these three are interconnected, because whenever you are optimizing any bioprocess, this knowledge of synthetic biology along with these two engineering process will enhance your yield up to manifold, provided there is no thermodynamic limitation. So the thing is that how to do it? First of all, you can do all the in silico modeling, and for the in silico modeling there are various tools which are freely available at NCBS site. You sit quietly and open this site, and you will find out there are many tools, and using those tools you can perform studies related to your area. And after getting the in silico information, you will get the input that what new you can do for improvising your process, and that improvisation involves the knowledge of genetic engineering as well. Let me tell you, if you are looking at it, that whether you are working with the DNA or with the RNA, whenever you isolate RNA, you go to the reverse genetics. That means what is the current state of expression of the genes in the form of messenger RNA, so that it is reverse synthesizing, you are reverse synthesizing its DNA form, and that DNA is then used for amplification or for identifying any of the unique genes among the community, of your samples I am talking about. So if you identify those genes, so those new genes can really be more proficient in terms of the process optimization, and those genes can be fetched out and can be integrated in the genome of your microbe of your choice, with which you are at ease to handle with respect to different aspects. And you should know here that the success depends upon the design of the specific primer, and the designing of the primer must be done very judiciously so that you are not going to miss any of the sequences which is of your importance. And for that you have different tools among different gel electrophoresis techniques. The denaturing gradient gel electrophoresis is very important to give you or identifying you the unique genes among the community. Similarly, you can also identify the metabolomes, and those metabolomes can give you the identification of specific metabolites, and that is responsible for successful or for success of any biological process. So these transcriptomics or metabolomics have the future of any process optimization. Any, I'm telling you, whether it is therapeutics, whether it is diagnostics, whether it is industrial oriented, okay. And for identification of these, we have different type of techniques, among which fish is very important. But you might have listened the lecture of Dr. Ray, where the sensor based techniques nowadays is gaining the importance of identifying a specific sequence from the crowd of the sequences. One can obtain it. So it is just like a gold sensor is being used to find out gold from the rocks. Similarly, these are the nano-sized single-stranded DNA sequences which can find out the complementary sequences as you have designed the single strand over your substrate or over your sensor. So here also your specific nanoparticles which have the fluorescent or luminescent properties, they will play a miraculous role. But one has to think in that direction, that what one person wanted to design. So depending on that, you can really engineer your experiment as per your need. Earlier you were engineering your cake, decorating it with different piece of chocolate or fruits or nuts, whatever. Likewise, if you suppose your cake is your strand of DNA or a protein and you have to engineer it, yes, now with the advent of those modern technology instrumentation techniques, we have entered into the era where a single nano, you say that 2 nanometer changes in dimension or few picoampere change can be noticed, and those change is, if it is if it is recorded, that means it has the possibility of exploitation. Look at the forensic science. All these technology will change the overall scenario of the, this is the generation wise change. You might have heard first generation technology, second generation technology, third generation. Similarly, so now we are changing the technology so fast. Even in this COVID situation, this molecular technology of the quantitative real time PCR has only led us to know whether the patient is suffering with the COVID or suffering through some microbial infection, any other infection, or suffering through any metabolic disorder, and so on. So you have to realize here at that moment that how these technology, whether it is associated with the isolation of the RNA and synthesizing the complementary DNA, which is giving you the overall expression profile of status of your microbe or your ecosystem or your health. It's up to you where to and how to exploit these technologies. Now, I think whatever I wanted to tell you, beside that biohydrogen and biodiesel production, now it is your interest. You can extract the knowledge from me. It is up to you. Participants, now you can ask any question if you wish. We still have some time, and don't worry for the time. We can extend it also. Just try to get all the information. Any query?
Ma'am, I have a one question.
Yes.
Good afternoon, ma'am.
Hello. Speak loudly.
It is audible, ma'am? Now?
Yeah, now audible. Yes.
Okay. Ma'am, means if you are means we are planning for a larger scale, let's say means around 1000 liter or for integration process like algal system and microbial system for the production. So what design we should means keep in your mind while designing the
Okay.
reactor?
Okay. You wanted to produce biodiesel? What do you
Uh,
want? Yeah,
means
or
biohydrogen
integrated. No, no.
or
The
biohydrogen products.
biohydrogen. Okay.
Yes.
Then why don't you extract oil from the algae also? Because it
Yes,
has
ma'am.
lot of
Means
oils.
after the means once we produce the hydrogen, then we can use the algal part for the oil means next step. But I want to know how the integration reactor design we should think of means while simultaneously we can use the algal cell and microbial cell for the production of
Okay.
hydrogen from waste.
Okay. You are from which institute?
I'm from Kale University, Andhra
Okay.
Pradesh.
Okay. I would suggest that are you using any biological waste as a feedstock?
Ma'am, previously I work on crude glycerol and wastewater for the production of means all biofuel like ethanol and hydrogen. Also, some
Okay.
part I work.
Okay. So
But
what you
for it,
do, I will
uh,
suggest you some plan.
yes,
You
ma'am.
use the wastewater for growing first the algal cells. So you will
Okay.
reduce the organic content. So
Yes.
for that you have to know the initial characterization of your wastewater and then grow your algal into that, then extract the oil from it. And then you digest those algal cells and then pre-digested material, do the pre-treated one and then put in the reactor itself along with the supplementation which I told you by optimizing.
Okay.
And then you in put your seed microbial culture into it, and that seed microbial culture will give you the good amount of hydrogen. I'm telling you, very good amount of hydrogen, and that you can do in different stages. In stage 1, stage 2, stage 3. So there will be the integration of the biodiesel and biohydrogen production both.
Okay,
And
ma'am.
for that, I would, what would be the capacity you would like?
Ma'am, initially I'm planning for in the lab, right? In the like normally we
Okay,
use the
for,
bottle like 1000 liter bottle or 2000 liter bottle.
okay, okay. What you do for growing the algal cell, you use the airlift reactor.
Okay.
because that gives you good amount of cells.
Okay.
Because what you do now, you use the waste, so it will be for help for everyone. Because all the food industry, they are just throwing all the waste. So use that waste, maybe distillery water, maybe starch water, or maybe cheese whey water, and then grow it, collect the harvest it, and then just pre-treat it, and then use as a whole, and then put a robust seed culture where you should have the spore forming bacteria and non spore forming both, and select the bacterium culture as that few of them are thermophilic, few of them are mesophilic, so you can operate it in both the conditions. Your culture will be alive throughout the season. Okay, and then wherever you do in the closed chamber so that temperature could be maintained. So without having any external jacket or water circulation system to maintain the temperature, you can get a good amount of hydrogen, or in a turn we can say good amount of substrate conversion. So when you will characterize at before the experiment its total sugar, and after the experiment its total sugar, you will find out there is a marked difference. There will be a marked difference. So conversion rate of the substrate you can obtain. So at the end you will find organic acids, maybe majority acetic acid, some butyric acid, some propionic acid. Then try to distill all those, report the yield, and then report the yield of the lipid which you extracted, and then convert that lipid into biodiesel also. And as well as here you report the yield of biohydrogen, or maybe if you are getting biogas due to the mixture of the microbes, then also it is equally good. You can put any scrubber in between the collection sample. You can also get the pure hydrogen. And it will be a very nice study, and that you can publish. And I will tell you with my experience, you can go in different stages. Because what happens once it pH acidifies, pH goes down, hydrogen production suppresses.
Yes.
In that case, you change the medium half and then put new supplementation. And then you will see that immediately lag time of hydrogen production will be reduced. And immediately you will have good amount of hydrogen. If you want to go for the continuous reactor, then you can maintain the pH. And for maintaining the pH in situ, I would recommend you go for the co-culture. Because this I have tested. The two bacteria, one which is utilizing your substrate and giving the acid production fast, and the other one is utilizing the acids and giving you the
Hydrogen.
yes. That co-culture, like Rhodocynomonas palustris, it utilizes like that. Or if you give some little light conditions, then co-culture of photofermentative and fermentative will give a brilliant result. And the problem of lowering down of pH is also eliminated. Now it is up to you whether you
Ma'am,
want a batch culture or a fed
ma'am,
batch culture or a continuous. Yes.
ma'am, in co-culture, if I am giving light, so that means you will give continuous light or in half phase, or how will?
No, no. In that case, you have to give the continuous light.
Okay.
It is not the LG, na. You have to give the 16-hour and 8-hour. It is up to you. If you want to go in the night, you can make it switch it off also. It is no problem. Or if you want to go for the day culture, then go for the open one.
Okay.
In all the cases, you will get a good result. Just you have to take care of the pH. You have to take care of the some important cofactors like nickel and iron. And then definitely you have to take care of the initial COD.
Okay.
That is why I always say that in the seed culture, if you are not going for the pure culture, seed culture must contain some of the bacterium which produces the cellulase, which produces the protease. So your initial substrate gets hydrolyzed. This is the success of your work. Okay.
Yes, ma'am. Thank you so
And
much.
then you optimize it also
Yes, yes.
before going for large scale. And that optimization you do through the RSM. Okay.
Okay, ma'am. Thank you so much.
Any other question? नहीं पूछो, किसी को कुछ पूछना है?
Participants, if you have any query, any doubt, or any suggestions that needs to be taken into consideration, please, the platform is yours. Kindly come forward.
Let me tell you, this is the last practical. Tomorrow we'll be having case study.
Which one is
And
the
then
best category is used in immunotherapy?
a talk from art of living expert, Varun Upadhyay.
Ma'am,
Okay.
there is a question. Which one is best catalyst is used in microalgae biodiesel production? Which one is best catalyst to be used in microalgae biodiesel production?
Hello. Usually, I told you that enzymes work at the ambient condition, but it is very costly. It is safe also for the environment in terms of sustainability. But when we talk about the results, it is always the acid catalyst which gives a better result. But then it may lead to the charring problem. So sometimes it is avoided. Some people recommend the base or
That's it, ma'am. Participants, this is the last call to address your query, because tomorrow you won't get much time. Tomorrow we'll only have one session of case studies, and after that we'll wind up with the faculty development program. So again, if you have any query or any suggestion that needs to be taken into consideration, kindly come forward. So I guess that's all for the day, and now we are closing our session on the day 4 of this faculty development program. Tomorrow is the last day for this workshop, and we will meet soon at 10 a.m. followed by a yoga session, and then feedback evaluation, and we'll end up with the valedictory function. So I think you all are allowed to leave since we are over this session. Thank you.